So what this tells us is that even though they carry a genetic mutation, there are other genes floating around in the background, the cumulative effect of which can completely negate this indisputably bad gene. And what we do to find all these other genes is a series of crosses to map them. So I take this white mouse, I can cross it to another this gray mouse, cross it to a black mouse, or to a white mouse, or to another gray mouse. And what we see is in each of these crosses, all the mice have heart defects. So what we're looking at is really like a poker game, where genes are dealt out like cards, and the combination that you get is either a good hand, a so-so hand, or an incredibly bad hand, right? We've taken this particular cross, where a gray mouse is crossing another gray mouse, to map the genes that are involved. This is called genetic linkage analysis. So very simply, this cartoon portrays the two copies of a gene that you get from your parents. So the white mouse gets white genes, the black mouse gets black genes. Your genes are lined up on chromosomes. Humans have 23 pairs of chromosomes. Mice have 20 pairs of chromosomes. Suffice it to say that I can, in the lab, figure out whether you have a black or a white gene. I won't go into the details. And what we want to do are find mice, all of which that have holes in hearts, that all have, say, little v. So big V, say, might protect against heart defects. Little v, when you get two copies, causes a heart defect. We do this across the entire genome. So there are 20 chromosomes. I don't show the sex chromosomes and the number in the mouse from 1 to 19. What this is a plot of the odds that we are on to something. So geneticists think a bit counterintuitively. What we do is we assume that we have the null hypothesis. We have nothing. And then on the chromosomes, what we look for are mice that have all two little v's. And we think about what the probability is that if there were nothing, that all the mice would have two little v's. When I was trying to figure out how to explain what are called log scores to people, it's really hard to do. So I realized, in keeping with the poker analogy, that what you should do is just think about poker. Geneticists think a log score of 3.5 is pretty good. If you found mapped locus, so a region of the genome with a log score of 3.5, you're probably onto something. A log score of 3.5 in poker is equivalent to pulling 4 of a kind. A log score of 5 is equivalent to pulling a straight flush, and a log score of 6 is equivalent to pulling a royal flush. So it is highly likely that we've found something on chromosomes 6, 8, and 10. Years ago when I started this project, the project would have died because, and this is where all labs that do genetic linkage analysis, at least in the 20th century, have died, because even though you map it to a region of the genome, it's still really big, and there still can be dozens or hundreds of genes. But now all we have to do is go to a website, see all the genes that are there, and then we can develop experiments to try to figure out which of the genes might be interesting. I can tell you from all the defects that we've looked at that it's highly unlikely that any of the known cardiac developmental genes, the genes that I think are not going to lead to any drug, sit in these regions. So that's good for me. We're going to develop discover new genes that might be targetable. How do we get to plan B? So the few defects I've shown you, aside from hypoplastic left heart syndrome, are actually incredibly easy for cardiologists to fix. So we're really good at closing holes. And conceptually, that's not so hard, right? But if we have to change the plumbing around or put a new piece in, like a new valve in that was never there before, that's really hard to do. You can think about plumbing in your house. You can fix leaks, but if you had to change the plumbing in your walls, you have to rip the walls out. It's just not going to happen. And so these technically challenging defects are actually pretty rare. And that leads to another interesting question. If you get away from thinking about causes of disease, why is it that technically challenging defects are rare? So they're rare in the general population, but they're actually quite common on the 7th floor of St. Louis Children's Hospital. It fills up the whole floor and keeps a dozen doctors busy. But they're actually pretty rare. If you believe that a combination of genes is important for the development of a heart defect. There are two possibilities. One is that there are so many genes involved that the probability of pulling a royal flush is really small. So it's just not going to happen in general. The other possibility is that there are few genes involved, but their effect is weak. So what I can do, this is an example of an atrioventricular canal defect. It looks like a fish heart, where there's just one valve shared between the left and right side. This defect is commonly seen in children with Down syndrome, is we can do genetic linkage analysis. And so this is actually pretty exciting for us. What we see was that we pulled the equivalent of now almost a 4 of a kind, maybe a 3 of a kind. On chromosomes 3 and 5, it's highly likely that we found genes that are really important in the development of this uncommon, technically challenging defect. In these, when all the mice have the same cause. And if you look at the versions of the genes that they obtained from their parents, it's actually pretty interesting. So these plots, I'm sorry, are a little bit complicated, but bear with me. All the normal mice are shown in the white bars. I told you that you can get one of two versions. So all these mice here have 2 C's, these mice have 2 A's, and these mice got either a C and an A, right? All the normal mice have what's called the expected Mendelian distribution. It's as if the parents were giving one of their 2 copies at random to their mice, to the babies, to the normal mice. On the other hand, if a mouse has this rare complex defect, basically, if a mouse gets 2 C's or 2 A's, no mouse will get a heart defect, or only one of the couple dozen that I looked at gets the heart defect. This is pretty exciting because I am never going to be able to fix the genetic mutation that causes this defect. But if I could develop a pill that mimicked the activity of CC or AA, maybe I could give it to a mother as she became pregnant before the heart had formed to prevent this bad heart defect from forming. This is not such a crazy idea because mothers now, as soon as they realize that they're pregnant, they go to their obstetrician. Their obstetrician prescribes folic acid, a type of vitamin, to prevent neural tube defects, like spina bifida. So this paradigm actually already exists. And so what we can do is, if we understood how bad defects form, and if it's as simple as a few genes, we can figure out what they are. I know that they're not known cardiac developmental genes, and manipulate or try to mimic them or inhibit them to prevent heart disease. So that's plan B. These genes, I think, are probably related to how blood flows through the heart. Cardiologists have this intuitive understanding that if blood does not flow through a certain structure during development, it does not grow. And blood flow is something that we understand really well and can manipulate. So there are medicines for high blood pressure. All those drugs affect blood flow. There are drugs that affect how stiff the heart is. That affects whether the blood flows to the left or right side. So I think we're going to find those types of genes, and those are things that are really easy to understand for us and easy to prescribe. There are many drugs for those types of problems.